Okay, so we talked about, Connie's introduced how we get sequence data. We've started to talk what does genomics data, what does sequence data files contain, and what do they look like? Okay, and this should give us a good understanding of what we're actually dealing with when we start doing some of the programming and coding later on. One of the first things you should absolutely do with your data is try and find a way to visualize it in a linear genome browser. Take your signal bigwigs, take your beds, and just see how they are arranged. Where is the signal across your data set in something like IGV? So on this page, we have the next course. It's IGV, it's here, same setup. If you want to set up IGV, I think we have download instructions here. I'm also going to talk briefly about it. And then we have our one little presentation to go through. I've got, I think, 40 minutes, Matt, is that right? But it shouldn't.
Yeah, 40 minutes.
Great, thanks, Matt. So it shouldn't be a rush to get through this. Okay, so I'm sharing my whole screen. Okay, so what I'm going to do is actually, I've got my presentation on the side here. To avoid me hopping between presentations and IGV, which I want to have open, I'm going to start with the presentation and then I'm going to flick to IGV, but I will be following along so that we're doing it in the right order as we go. It's a little hard to navigate Zoom multiple things. Okay, so let's get started. So IGV, and just as an overview, talk about what it is, how we navigate around IGV. So what do all these panels mean? How we can start to load and actually visualize some data, things like adding sample information, genome information, annotation, and then finally, how we can customize some of this visualizations, how we can get our data arranged and visualized in useful ways. Okay, so this is really just a crash course, but hopefully we'll show you enough to start to really use this effectively. So IGV, I mean, there are a few competitors for genome browsers. Competitors, maybe they're allies, I don't know. But there are things like IGV. I think UCSC is a very popular browser as well. And then we have things like the WashU browser, IGB, Zendu, lots of complex genome browsers. This was created by the Broad Institute, so it has great caliber. And as a genome browser, it allows us to visualize genomics data, things like expression, our ChIP-seq, our resequencing, multiple alignment, shRNA-seq, within the context of a linear genome. Okay. Handles most common genomic data types. So IGV is being actively followed, maintained, developed as we go. I'll show you at the end. Their direction is moving somewhere else now, but we are well covered in this. As opposed to some of these other applications like UCSC, this is a desktop application on your computer, and it runs in Java, so you'll need Java in order to run it. Most of us will have this. By being a Java desktop application, it is not dependent on a server, right? So we can load data locally or from a URL, and it's going to consume the memory on our computer. So if I want to open 100 files, my computer might start to slow down depending on the size of those files. But the other advantage of being local is I don't need to worry about uploading my data to a server, both privacy issues or even just the speed and efficiency of this. I can review my data very quickly in my own computer. And as you'll see, it really is very fast just to get an idea of your data very quickly. So like I said, it requires Java. If you want to get IGV, and you'll need it for this course, you can follow our links, but you can also follow this link here. And that will take you to a download page. Maybe I should open that page. Oh, wow, this looks a little different now. The download page, it knows I'm on Mac, so it's saying download the Mac app. And actually, this includes Java, so we don't need to worry about Java anymore here. You also have things like the archive versions, and these are incredibly useful if you find that your new version of IGV doesn't have a feature that you really wanted in your display, in your image. You will find the archive versions in this place here. And this just means you're never going to have to worry about losing this functionality. I'm going to go to this at the end. There are advances on IGV, including things like interactions with Jupyter Notebooks, JavaScript versions, and what we'll have a brief look at is this IGV web app. So IGV no longer running in Java, but running in your browser locally, not on a server, on your own browser. Yeah, download to your computer, it runs locally, archive versions available. I didn't see this yet, but there is available a web start. So we use the web start quite often. What's nice about the web start way to launch is that it's always going to run the latest version of IGV. What is a pain about the web start is that it's going to run the latest version of IGV, and you may have been comfortable with your previous version, and that was set up in the features, and the tools were all in the right places for you. Okay. If you're using this web start, it's always going to go get the latest version. Okay, if you want a solid, this is the version I'm working on, you should download the self-contained version. That version with Java would be the one. Okay, so now I'm going to start to look at IGV. I'm going to hop, I've got my slides on the left here, so I know where I am. But I'm actually just going to open up IGV, and I can talk through with IGV in front of us rather than maybe a picture of IGV. Okay, I can get rid of this. So hopefully you can all see, I think I'm sharing my whole desktop. Oh, hang on a second. And I'm going to make sure I'm handling my whole desktop. There we go. Okay. So hopefully you can see IGV here. I'm going to clear this out so we have a nice fresh IGV. Okay, so this is what IGV looks like. This is the GUI. Actually, if I make it a little easier. In things like Mac, you're going to have the actual GUI sitting in front of you, and then on the top bar, you're going to have some of these menu options here, genomes, view, tracks. But it's in here that we're actually going to see the data. So talking through our various parts, we have our first part here, which is our sample information panel. So once we start loading data, it's going to give us names of our files here. Okay. We have our genome navigation panel at the top here, and it's here that we can see first off the whole chromosome we're on. If I was to come out, I'd be across all chromosomes, but we can see them spread out across the top here. And it's with these panels, I can jump to various regions of the genome and go review what I found interesting. And then these two panels at the bottom are very similar. Sorry, I was going to try and make that a little wider. Yeah, it lets me. So we have what they would term the data panel here, and I would say this is your data. So the data that you're trying to review, your bigwigs, your BAM files in IGV would sit in here. But on the bottom would be your attribute panel. So these are more genome reference tracks, such as our gene models, which you can see here. And that would kind of sit on the bottom. So our sample information, our genome navigation here, our actual data we really want to review here, and then our attributes are things like genome models we want to compare alongside in the bottom. Okay, so we have our menu bar. At the top here, we have file, and that contains things like how we're going to load our data, old sessions. We have genomes, which allows us to deal with loading and managing new genomes, so very important. We have view, which we'll look at later. This allows us to customize the preferences. Tracks. This actually really just allows us to do things like resize the size of the track in our window. With some group information, we can do some filtering on our tracks, apply filters by their name, for instance. Regions allow us to define and review regions. And we have this tools, which is really a fantastic addition to IGV. And this contains just some tools to allow us to review the data a bit. It might be a tool to compare a sequence to the rest of the genome or to find a motif in there. Okay. So if we want to move around IGV, it's actually really simple. We've got a few different ways of doing it. We can either select the cytobands, so chromosome 1, 2, 3, 4, 5. We can select these. Once we're in a genome, we can select the actual cytoband we want to look at. Zoom in that way. We can also scroll, and we can say like this. If I select the bottom here and I hold my mouse down, it's going to allow me to navigate so I can review across my genes. I think if I do left and right, it can be like a computer game. Not sure what up and down does. Okay. So the zoom is very easy. We just select a region and it will zoom in. If I want to zoom out, I can do this part here. Scrolling we just saw, and then jumping to regions. IGV is kind of cool like this. I can jump to regions based on my chromosome names here. I can also actually provide a region I'm really interested in, like that. Or because I got my genes track here, if there's a gene I'm really excited about, I have my menu here and I can go and look at actin beta, which is in the middle here. So easy enough to navigate around IGV. Once I've found my region of interest, I might want to just bookmark it, as they say. Mark it as a particularly interesting region, either for other people to come back and review in the future, or maybe I want to extract something from that region using the tools in IGV. Cool. So in order to bookmark the visible window, whatever I'm looking at now, it's very straightforward. Just need to go up to my regions and I open up Region Navigator. And if I want to, I can add the current region just by doing add. Right. And if I wanted to as well, I can add a description to this. My favorite region. Okay. Now if I zoom out, oh, it's already happened there. If I zoom out on my track, you can see it's now highlighted this region in red. Okay, and I can actually, if I close this and click this, I can pull up a menu. And I can start to mess around a little bit with this region. Okay, most usefully, I know where my region is. It appears in that top bar there under red. Okay, I think on top of just selecting the region, I could also use this. So if I really want to get a little bit more fine, if I delete that now, if I want to get a bit more fine detail, I can come in and I can use this define a region of interest button. And I can just select here, select up here. And now I've got my new region of interest. Okay, a little bit more refined maybe. So really useful, I think one of the first useful things I got out of IGV is if I wanted to then find out what the sequence was underneath this region of interest, here I'm just selecting randomly. It might be a peak, it might be a particular bit of signal you've seen. I can just go and copy the sequence directly. If I have a text browser open, oh, I got some example sequences here. Should just be able to paste it there, and you can see I've got my sequence for whatever was underneath my region at this point. Okay. Another really nice thing we can do, which is obviously very useful, is BLAT this sequence. So BLAT is just a very simple way of finding an alignment of that sequence to other places in the genome. Does that sequence appear in other places? Right. And this might be useful that I want to investigate how unique this region is. And then I can go through and actually review how well it's aligned, what gaps I have. Okay, we can see the alignment now is appearing in our attribute track, right? Because it's not really data, it's an attribute. Now I've lost my thing behind. Okay. So it's a nice little feature within IGV just to allow you to quickly select a region, save it, but then also find regions in the genome which maybe look similar. If I want to extract those actual regions, I can click here and just export features, and we can do this for anything. Export features, and it's going to give me back visible data, but it's actually going to give me back my regions. A bed file, we know these are just chromosome start end. I can retrieve that, and then I can use that back in R or in other programs. Nice feature, and we're starting to see it here, is delete. If I zoom in, I mean, actually, if I zoom out a bit, we can see we have a representation of all the sequence here. If I keep zooming in, I can actually see what the sequence is itself. I should be able to reverse the direction as well. So we can actually see the sequence. We don't need to just rely on it or retrieve it from our table. If I can right-click this, I can show translation as well. So if I wanted to, I can actually look at what the protein is associated to the transcript if I translated it. And then I think I have some other features, like I can flip the strand, or I can change the translation table. If it's not a standard translation table, there's different codon usage. I can also select that here. On top of reviewing just unique areas, a really cool feature to quickly review your data. Maybe we should load some data. Have I even got to that part? No, we can wait to load data. Is gene lists, right? So we have a region navigator. I can create one region. That's useful. But actually, maybe I want to review several regions at the same time in this window. Okay, so we can have lists which are pre-built inside IGV, and they've got things like KEG, example one. So if I select maybe one with a few in, you can see what happens here. If I select this and I say I want to view it, you can see it puts all the genes or the signal over these genes side by side, right? And I can actually see. I should probably load some data quickly. Desktop. Just going to load some data to us to review quickly. You know, I can get an idea in one panel straight away. This one and AKT2, those are very highly expressed. There really isn't much signal in the other ones compared to them. So IGV allows us just to rapidly review data over multiple regions in just one or two clicks using this gene lists. Can actually, I don't really cover this in the slides, but you can obviously import your own gene list too, or you can create a new gene list here. Just need to paste in the name of the genes going downwards one line at a time, or the locations you want to go and have listed here. So very easy to create your own gene list as well. Okay. So the next thing we're going to look at is loading data into IGV. I've created my data. How do I actually start reviewing it? So I can clear out this data first. Remove track. Okay. Maybe clear this out too. Okay. So the first thing we're going to look at is loading genome information. Okay. So load genome information we have. First off, we have lots of pre-built genomes, and I think some of these are custom for us because we have a few strange genomes we work on. But lots of genomes available to us. If we wanted to do something a little bit more custom, we can actually go into the genome here and go load genome from server. That's one option. And they've got lots of genomes pre-built. And if I just select here, I can go OK. It's going to download that version of the tomato genome for me, and then that will be available in this dropdown. Okay, so these are just the ones we're currently kind of working with. Important thing here is if I click download sequence, I'm going to download additionally not just the limits of the genome, but actually the sequence information for the whole genome. So you can imagine if you click that, it's going to take a lot longer to download the genome to your computer. If you do that, you can still visualize the genome, but you won't be able to see the sequence underneath your regions of interest. I don't need to do a new genome here. Okay. If you're really a very custom genome, you can actually create your entire new genome. All we need is two files we actually know. A FASTA file, in fact, we don't need the other file, but you can have a GTF or a GFF here. So those are our gene models. But all you need to do is have a FASTA file, you give it a unique identifier, a description, say mosquito, what version, and then you just pass in the FASTA file and it will create a genome and allow you to review data over the top of this. Okay. This gene models here, we can also import new gene models. We just need to go file. We can just load from file and load any gene models as a GTF, and it will be loaded in this track here. So actually loading sample data, we do in a very similar way. We go file, load from file. As I said, IGV really covers a whole range of different data formats. So I think I had a link here. There's a whole range of different data formats. This is always expanding, and you can see pretty much a full list here. And this includes things like SAM, which we probably wouldn't want to load, and BAM, bed, bigwig, and big bed. I think TDF is, no, TDF is there one. I guess Tabix is, I know Tabix is supported. I don't actually see it here, but Tabix is supported too. Okay. So most of the common data types we can import and manage in IGV. They do have some of their own types of data. In particular, they have their own equivalent of a bigwig, which they call a TDF. Both created at the same time, both written up and published at the same time. I think bigwig just had the advantage over TDF. For whatever reason, it became more popular. We can also include alongside our files some sample information as well. So we may want to have slightly better organized IGV. We can include things like the name, maybe the data type, the antibody which was used. And this just means that with this information, we can group parts of our data together, perhaps by antibody. It just allows us to visualize our data easier when we have hundreds, maybe hundreds of files. Yeah. So actually, it's a bit of a pain to set up this metadata sample sheet required for IGV. I think if I follow this link, it's going to send me to what it kind of looks like. This is their example. It's a bit of a pain to set up. So what we did is we actually wrote up a package called track tables. I did this actually in my old team. So this just allows you to create some of the sample information. And we'll show this in the courses themselves, but it allows you to create these files from simple data tables and also the XML, which allows you to load these into IGV. Okay. With that information, we can do things like sort and filter our tracks. We can split the tracks by group. And I'm just showing you that here, where we could take the data type. That was all unique. But as soon as I grouped it by antibody, we had our EBF antibodies here, our H3, K4, ME3 here, K9, H7, RNA pol2 here. Okay. So I think I had at some point an actual example of what that kind of looked like in IGV. Load from file, not there. Desktop. Not entirely sure I kept it around, but maybe I did. Oh, so those are the genomic files. I'm actually going to load in the sample sheet, which is an XML. Sorry, I haven't got it available on my computer, but I can perhaps show you that later on. Okay, so on top of loading data locally, which I think I show in a few lines, we can actually load data directly from a URL. Okay, so that's really useful. At the moment, if I want to load a file, I've got to have the file somewhere on my computer. Here I've got some bigwigs. I could just load these. Maybe I'll choose a slightly more public data set. Hopefully IGV should do this very fast. You can load data in from local files very fast indeed. So that's full genome data for something like 12 samples. Came in instantly, and it came in instantly because it's a bigwig, so it's very computationally efficient to actually allow us to load it in and summarize it. It won't let me go to chromosome 1 for some reason. Okay, so loading from local files is really easy. If I wanted to load from a URL, that's also pretty straightforward. You can just give the file URL, HTTP in here, and then it will be able to read this and load this in to IGV. Okay. What's really cool is that if we use things like bigwigs, because these contain an index, it doesn't have to download the whole bigwig to your computer before it starts visualizing it. It actually just needs the portion of the genome which you're looking at, and it will just load that bit in. For BAM files, and if you remember, BAM files required this .bam was the file, but they also had this .bai. This is the index associated to the file. If we want to read from a URL for a BAM, we also need to provide the index. And that's because it's going to look in the index, work out where the information is for the window we're looking at, and read that back in. Okay, cancel. I'm not going to load anything in now. Where are we up to? Okay, so the next thing I think we're going to look at is how we actually, so we can load from URL, but we've also got load from server. So that's good. Here we have annotations we can load in, things like gem code. This is different gene annotations we might want to load in. If I press OK, it may take a few seconds to load them. Oh no, not too bad. And now that's brought in not from any version or I have locally or in a URL I control. They manage their little server, which allows them to then have things like annotations, maybe SNP information. This is the information we had in VCFs. VASCOM, we're not going to load that. That will take a while. But this is the homology, the how conserved parts of the genome are. It says here zoom in to see features. So if I actually zoomed in, should be able to load some features now. It's going to take a little bit. There we go. So that's the gen code actin B, which apparently has a much longer variant. I think I'm just going to change to a new, I think it's HG19. So there's a little bit more information available for HG19 in that they have up until this point, and I am assured that they will do this for HG38. They also allow us to load directly from ENCODE. So this, and I showed you ENCODE in our repositories earlier on, just before this. This contains now all that data we saw, narrow peak information. We have bigwigs. We have BAM files themselves we could load. We also have genome annotation like GFFs, so how they've defined their GTFs. Should be able to. Oh, CH12. And they don't have our, oh, because that was mouse, wasn't it? I should be able to select files, and if I want to, maybe something small, we could then load this into our genome. Okay. What's really nice is this is also carried across this sample metadata I was talking about earlier on. So I can probably load in a few of these. And here, load from ENCODE. Let's grab a few of these. That's probably a stupid mistake. It would now take a long time to load this data. But great. So now we have our peaks, and I can start to show you some of these nice things that I might want to group these tracks by this information. I want to group them by antibody. That didn't work at all. Tracks group by, oh, they're all the same antibody, lab. Okay, so now we can look at side by side. I can arrange this data by Stanford and Harvard using the metadata which they have attached to this ENCODE download. If you want to get this metadata for yourself, we have track tables, and this will allow you to build this from just a data frame with your sample information. And also filter out. I wanted to get rid of, I don't know, Harvard. I think I need to ungroup it first. But anyway, file, new session, we'll go back to HT38 for now. Okay. So, loading from a server, your own server, loading locally, using some of their pre-established data on the server, or getting access to ENCODE itself, you can do all through IGV. Okay. So now we got our data and we know where to get it from. We can start to visualize some of these common file formats inside IGV. And IGV is going to make some decisions about how it thinks we should display these. Okay, so I don't need to say I'm loading a bigwig, draw it like this. I'm loading a bed file, just make it clear lines. It knows what to do, and most of the time it's going to make a sensible choice. I want to put bigwig, make it look like bars or a graph, a line. Sometimes it won't, and we can still control that. So for information on what file formats and how they might be displayed, you can come look at recommended file formats in the IGV Broad websites. I don't really need to show this, but we looked at bed right earlier on. The computational equivalent of that would be big bed, right? So this is the one which would be compressed for IGV. This is very basic. It's very simple display. It's just tab separated, chromosome start end. We have big bed 6.2 potentially, which would have a name. And what we would get here is, as you would expect, just the block which represents the region, the line in my bed file. And then, because if I had a name attached to it, we would see that attached there. Right? There is probably names associated to these. It's just they would overlap, and IGV has decided to just show us one. So WIG files, bed graph, and bigwig, they all contain the same information. Right? So I could have a WIG file, display it in IGV, a bed graph, display it in bigwig. They would all look the same. But bigwig is absolutely the choice. You're not going to be able to load a WIG file or a bed graph into IGV in a memory-friendly way. And these look just like the one we had up, actually, when I was showing it now. This is how you can see these kind of peaks of signal over your regions of interest, or maybe this one looks really noisy, kind of blocky. Okay, so bigwig bigwig and IGV, it just allows you to look across your genome, nice linear genome, and see where you have your high points of signal, where you have the high scores stored in the bigwig. So BAM files, these are our alignment files. These are going to contain a lot more information than our bigwigs or our bed files. Okay, so these contain both the read, the sequence, the alignment. To load these into IGV, we need this .bai. I think it may let you do it, but it's definitely going to complain if you haven't indexed this file yet. You can download these indexes when you download the files. We're going to show you how to create the indexes. And what IGV does here is it just displays along the bottom bit here your actual reads. You can see every single block you see here is a read. And on the top, it does something kind of neat as part of the process in IGV. It actually builds a coverage graph for you. So it translates these individual reads. And for every base pair we're looking at, it's telling us you have this many reads at this particular position. Right, so this is probably, for me, often this is more useful than seeing all the reads here. It's too much information. This is our summarized coverage. So IGV allows for customization of the track displays. You can actually just select view preferences, or you can select on the right-hand side of a track. So you can go, if I load some data, load from file. Okay, I can do right track. I can do some stuff here to change the appearance of my track. I've also got this view here where I can change more general parameters about how IGV is actually configured, how it looks. So in general, we have some really useful things. I'll go back to IGV to kind of show you these in action. In general, we have some useful things like relative paths in the session files. When we save it, maintain it as a relative path, show the attribute panel. Really useful is when I go and zoom in on a region, how much flanking space do I want around that region? Okay, if I zoom in, you may notice my IGV, we'll go there in a second. No, I can do that now. My IGV, if I go to a particular location, I'm actually quite far away from what I wanted to look at, right? Actin beta's here, and I have a large area flanking around it. If I go to my view preferences, I think this is where I've set it. Yeah, here we go. So I've set this really big. This because at the time I set this, my laptop can handle holding a wider area in display, but also it allows me, if I want to look at regions around that particular peak, I want to set that wider. If I can set that again to 100, it's going to be perhaps not so useful for me, but we can see it in action. Okay, now I just get really zoomed in on this particular read. Maybe that is useful. If I was using RNA-seq, this is probably giving me a slightly better view. Okay, I can hear my computer starting to struggle. Okay, we can also control things like the heights of our tracks. So we can say how many pixels do we want for a track. It's the default track height. So this is just going to control preferences, tracks. I'll make this BT 90, and I make this 50. You just see that my track will get wider in my screen. I can also configure that here, change track height. But this would then set it for all of our tracks. And I think one of the really useful ones is alignments. So there are lots of options here. Maybe I should see if I've got, I'm not sure I've got an alignment track, but if you go to preferences and we look at alignments, this is really worth looking at. So when we saw our slides here, you can see that we have a coverage track and then we have the reads underneath. Okay, maybe you just want to use your BAM to see the coverage. Maybe you really want to look at your reads to see the sequence within your reads and how that relates to the genome. So here we can display, choose what we want to display. So if I want to see the alignments themselves, I can check that on and off. If I want to see coverage, signal above my reads. And then we also have the option here for junction tracks, which I think I talk about in a little bit. And that's really for splicing, right? When a read splits across exons, such as from here to here, if we have a BAM file, we may want to actually display those in particular. Importantly, you have things like downsampling reads. So by default, I think this is turned on. So if you look at the number of reads in IGV at a particular location, it's actually been downsampled based on these parameters here. And finally, a really useful one is the visibility range of BAM files. So if I wanted to go and start looking at BAM files in IGV, just as we have, how far around your region do you want to look here when I zoom in, this tells me that I can look in BAMs, I can look at my BAM files within a window of 300,000 KB, 300 KB. But if I start to zoom out more, my BAM file will no longer be visible. Okay, I'll try and load up a BAM file at the end just so we can have a look. Okay. Okay, so the next thing is we can do some, really, I think the next things we're looking at is finer control. So actually changing some of the appearance of these tracks. So whereas view preferences is going to change globally, I load some more data in now to make my life a little easier. I'm going to be a little bit late, I'm afraid, folks. Open them. I'm going to scale them. I'm going to show what I'm doing here. Scale. Okay, I can start to do things like change their color very easily here. I can make certain groups one color. I could maybe, if I had negative scores, I can also select track colors negative for when they're negative to go a different color. I can also change the graph types. So this really is dependent on your preference, but if you wanted to, you can make everything into a heat map. But rather than being, by gene, it's now going to be across the genome to see where your signal is, or perhaps you want something like this, or maybe just something more minimal in a line plot. We can control that all here just from this right panel. What we have. A really important feature, actually I'm going to show this in the slides rather than, a really important thing to consider when you're doing your visualization is data scaling. Okay, so often you're going to normalize your data so you can review your data under the same scale when you look in IGV. Okay, we can control the data scale for one track or combinations of tracks just by using this data range right click on the left-hand side sample attribute panel. A really useful feature in IGV is something called auto scaling. Okay, it's useful, but it's also dangerous. What this allows you to do is select and say for a particular track, once I'm scaling it, just show me or adjust the scale so that I can see the biggest point in my track. Okay. So if we go look at that, what does that actually mean? It's easier to show you. I'll make these all our bar charts again. Okay. And I zoom out a bit. You can see we can't really see much else in this particular region. Okay, but if I zoom in here, we can see that the scale for all these tracks, all these tracks has adjusted depending on the height of the signal we're looking at. Okay, so if I carry on zooming out, as soon as I start to have a region in my window which was bigger, it's adjusted the scale, the size there of my track in order to accommodate for having larger signal here. So auto scaling is great when you just want to move across and see what signal you have in your region. But you don't want to be deceived by this being the same height as this, right? They are, when you put them side by side, they're just not. It's trying to fit it in your window to make life easier for us. So some more useful features about IGV. So I think a really useful feature about IGV is the fact that it actually allows you, like I mentioned, to see junctions. We can turn this on and off, but given a BAM file, it's actually going to show you when one read spanned from this exon to another exon. And this can really allow us and gives us confidence in how valid a particular exon change or exon usage is. Okay, so I think we use this in the examples as well, but here you can see in the heart, we've got an extra exon here, and we have an exon here. And actually you can look and see kind of these red curves coming into the beginning of this exon. And this is just highlighting the strength, the evidence of reads mapping from one exon to this particular like novel exon here and the standard exon here. Beyond that, you can actually do some kind of complex plots. One of them is called Sashimi, which comes from a different program called Miso. I think there's a whole salmon miso kind of thing there. But IGV, if you wanted to, with your BAM files, you can also take a picture and actually allow you to do a sashimi plot. Again, you're going to do this in the exercise at the end anyway. A really nice feature in IGV, which we couldn't do before more recent versions, was overlaying tracks. So I may want to look at one track on top of the other, and this is something which you can do in IGV. I may expect them to be very correlated, or I may expect them to differ at points, and I want to see the difference. So we can easily overlay a track in IGV just by doing this. Got 3 different colors. If I want to overlay them, I think I can just do overlay tracks. And we can start to see that we have these overlays, and I can see when my red sticks out from my blue sticks out from my green. And if I want to separate them out again, I can just do separate tracks. If I want to make it a little bit clearer, I can also adjust transparency to allow me to see what was really going on behind in the other tracks. Okay. We can also do things like a little bit of track calculus, and I'll show you this at the end. So we can use IGV to combine data tracks together. And we can do very simple things, not complex things here, but we can say add 2 tracks together, or very usefully is we can subtract the signal in one track from another. So if you have your test and your control, you can actually directly subtract them and then look at the differences in signal in IGV. Okay, so I can show you just doing that here. If I split these out, separate tracks, I should be able to go tools, combine data tracks, and I can do subtract, first from a second, that would go, and I get my new track here at the bottom. And again, I can then start to scale this and adjust this, and now start to review the differences in my data in this track, which I've just generated in IGV. Okay. Final thing, which I don't really use in IGV, but I know a lot of people do. Hopefully, we should be able to surpass this or tell you how to do this using things like R. But we have IGV tools. So IGV tools, again, is built-in tools, run IGV tools. And this has some functionality to allow you to do things like index your BAM file, right? So if I've just got my BAM file, I need to visualize it here. We know we can't do it without indexing, but IGV tools will allow you to do that indexing. A really nice feature, which I don't necessarily endorse because we like bigwigs, is the creation of these graph-like tracks, which are like bigwigs, but they're actually called TDFs, tiled data format. You can do that in IGV's tools as well. So there are a lot of people who take a BAM file and then they will process it in IGV tools within IGV, and they will get this new file, TDF, which is much smaller and easier to load in IGV. So I think the last things we wanted to talk about were like how we save what we have. You should be able to easily save the images which you generate from IGV and then take these out into your favorite Illustrator program. I think it's just File, Save Image, and then we can change the format to something like SVG, which I believe is easier to take apart in these external programs. Okay. Once you have got all your data together, once you've got your tracks organized like you would, maybe you want to share this with someone else, or you just want to save your session so you can open it up later on. You can do that very easily in IGV2. You just need to go File, rather than these sessions, you can Save Session, and you just give it a name, and it's going to save an XML file, and it's that XML file which contains, where is my bigwig, what color was my bigwig, what auto scaling was present. And then you can go off and pick up your session later on. Okay. So where else can you get help with IGV? Well, the Broad user guide, is very useful, the user guide. The Google Groups is incredibly active. I mean, because we wrote track tables and we work with them a bit. There was a point where I needed something fixed the next day, and the main author would come on and he fixed it almost in a few hours. So they're really, really helpful. And I think they're just fantastic. The last thing I wanted to show before we move on is that is IGV, but I mentioned IGV Web. So this is a really cool self-contained browser. So you don't actually need IGV. I think you can agree it doesn't quite look like IGV yet. It's not quite as bouncy. But it is pretty cool. It does allow us to navigate across genomes. And what's really nice is that you can load tracks from URLs. You can load them from Dropbox, Google Drive. But actually, I can run anything I could do in IGV just within my web browser here. And I can start to do everything at that point. Still, it's saved in an SVG, export it. I think this is where IGV is probably moving to. I don't think it's quite there yet, but maybe in our lectures in the summer or next year, we should be able to start to produce a little bit more details on what we can do with things like IGV Web. Okay, so sorry that went over a little bit. I think that's the end of this talk. Hopefully you've seen enough that you know you can load data into IGV. And we're going to do a lot of that over our courses, especially when we get to things like RNA-seq, ChIP-seq, TAC-seq. I think the next session starts, I guess, at 3, Matt. So I'm going to take a break, and I will see you back there.
It's 3 to 4:30, and it will be introducing bioconductor and sequence information stored in bioconductor.
Great. So I'll see you all there at 3. Cool. We didn't have any questions. Great. So still on our homepage. Done. Intro to R, plotting to R, tidyverse abridged, reproducible R. Covered genomics data very briefly. What do these data types look like in actual text? How are they contained for a computer to read? And then we can actually visualize some of these formats post-alignment. We're never going to be able to visualize a FASTQ. It has no position on a linear genome. But if we have alignment information, we have the position of the feature, we can then visualize this in IGV, and we saw we can visualize this with scores. And often this is our first thing we will do when we get our data back. We will try and get it to a process we can visualize.